Damian Menning is a research geneticist with the USGS up at the Alaska Science Center in Anchorage. His work primarily focuses on metabarcoding of environmental samples for community structure and diet analyses. He has recently started the mitogenome sequencing of endemic fish in Alaska. I know he spends a lot of time thinking about how to improve the reliability and repeatability of eDNA metabarcoding and the reference databases. His talk will provide what I guess we hope is kind of a 101 type discussion on eDNA metabarcoding basics. And he will hopefully try to, I guess, compare that or put that into context of what we usually talk about, which is targeted eDNA. So Damian, I'll let you take control of the screen and have at it. Thank you so much.
Okay. How's that look for everybody?
Looks great. I can see, it looks like you're in presenter mode and you're ready to go. Thank you.
Thank you, Adam. As Adam mentioned, I'm a research geneticist up at the Alaska Science Center. I've been working with eDNA since I started my PhD back in 2007. Back then it was called microbial ecology, and today you would call it eDNA. One of the interesting things that I found transitioning from microbial stuff to vertebrate hosts or vertebrate organisms is that there's a lack of a curated custom reference database like there is for the microbial sequences. So if you've done any microbial stuff, you'll know that there's the Silva database and a couple others. There's Chime and Mother and a couple other programs that can be used for microbial analyses, but there wasn't anything for just eDNA in general. So I've transitioned those, or attempted to transition those methodologies into genetic work that is applicable to the U.S. government. And I've been doing that for a couple of years now. So metabarcoding is shiny. A lot of people are wanting to get into it. This is just a cartoon from data I pulled yesterday from Web of Science. The first couple of papers that had to deal with metabarcoding started in 2011, and over the past 10 years, there's been a slight increase in papers published every year. So metabarcoding is simply PCR-based amplicon next-generation sequencing. And what that means is you develop primers or markers to target a specific region of interest that will give you a resolution on a target organism. Basically, you want to find an area that's going to give you enough differences genetically between other samples so that you can differentiate it. It is usually done in a two-step process. You can do this in a one-step process if you want. It ends up being a little bit more expensive in the long run if you decide to do it that way. And there are some problems with primer dimers and primers bending on themselves because they end up getting kind of long, like 40 to 50 base pairs, and you have to check for that, which can be done, but it does create some additional problems. So in the first step, you start out and you amplify your target sequence, and then the second step, you add on your indices. Now with an Illumina MiSeq machine, you have forward and reverse indices. So you can multiplex up to 384 samples in a single run very easily. So 4 plates of samples. And typically you get about 15 million reads from the V2 500 cycle kit. Now, here's some math for you. Even though you can do roughly 400 samples and you can get 15 million reads, you can also throw in multiple loci at one time. So if you wanted to look at the community structure of fish and you had your 12S and your COX-1 and Site B loci, you could throw them all on the same run and get enough data to validate your reads, whether if they're positive or not. But it does require some math. Typically, the acceptance rate is 0.1%. or 0.01% of the total reads passing filter for that sample. So if you're familiar with the MiSeq, you get a percentage passing filter for that run. And then if you multiplex your samples, you get a % passing filter for that particular sample. And so if you take that percentage and do the math, you'll get a number, and you can compare that to 0.01% to see whether the number of reads that you have would qualify as a positive identification. And this is typically the number I use. But if you're familiar with metagenome sequencing, they accept 10x coverage. So if you have 10 hits that overlap completely, that's considered a positive identification as well, or a positive sequence as well. So it kind of depends on what you're doing and what your question is and what your threshold is for acceptance. So why would you use metabarcoding? You can use it for invasive species, pathogens, community structure, determined species range, diet analysis, and eDNA comes from a whole bunch of different sources. Some ones that people might not think about are long-term storage. So museum specimens that have been stored for decades in alcohol are perfectly viable for eDNA analyses, especially diet analyses. I've recently published 2 papers on the bacterial community of gut microbes in shrews, and then also looked at the helminth species in that same environment. So it worked out well. We were able to do it. It ended up being a comparison of preservation methods, and all of them worked. So that's something to think about if you happen to have long-term storage samples. So the benefits of metabarcoding, especially in an eDNA environment, is that it's not invasive. It can be cost effective once it's developed. It's fairly simple to do. You can look at multiple species and multiple species level at the same time. And there's a lot of redundancy, especially when you use multiple loci compared to a traditional survey. And one of the really cool things about doing a metabarcoding analysis is the potential for added value and collaboration, which I'll get into more in a little bit. There are some limitations. The lack of sequence data that's out there prevents or can hinder development of custom reference databases. It can hinder primer development. When I was developing some fish markers a couple of years ago, there weren't any salmon species that were indicative or collected from Alaska, and I had to use some samples that were sequenced in Germany. It worked and we were able to get it done, but I was just surprised that there weren't those sequences available. So that's one of the things that you need to be aware of when you're developing primers and databases, that there might not be the sequences you need. Analysis methods can be problematic. You can use QIIME and Mother with eDNA. It takes a little bit of finagling, but it can be done. Or you can learn Python. Python's a programming language that is really powerful at analyzing strings of letters, which is all DNA is. So it makes it super easy to work with. The cost of initial development, if you don't have the sequences available, you need to go sequence them yourself, which can add to the cost of the project. But if you're working on common species, that really isn't an issue. So comparing QPCR to metabarcoding, which one should you choose depending on your project? It depends on how much information you're trying to get. This paper from 2016 compared QPCR methods to metabarcoding. And what they found is from collection through data processing, it took roughly the same amount of time and cost roughly the same amount of money. The plus side to this is if you're only looking for a few species, QPCR would be the way to go. If you just want to know a particular invasive species is there and you don't care about the community, QPCR would be great. If you want to know about the community but want to keep an eye out for invasive species, metabarcoding might be the way to go. So it kind of depends on what your question is on which method to take. But I've always liked the kitchen sink method where you throw everything at it and then see what happens and get a ton of data, and you can choose what you want to do with that data as opposed to only getting one particular thing. So how do you choose the primers, the number of primers? Ideally, you want to have at least 2 primer sets. One of the things that's always mentioned when you talk to managers about using an eDNA analysis is how reliable it is. Is the data valid? And if you use multiple markers, 2 or 3 markers, say both COX-1 and Site B and 12S, and you get positive identifications for the same species on all 3 markers in your sample, there's a fair amount of validity to that analysis. You can say, hey, we found this 3 different ways, it's pretty much here. So which one do you choose? Which genes do you look at? It depends on your question. If you just want to look at an entire fish community where you want to find sturgeon and salmon and trout, and you don't need to get into the nitty gritty, the slow evolving genes like 12S and 16S are great. You can get wide ranging communities from those markers. If you wanted to get into the nitty gritty, like which salmon species are there, which cod species are there, that's when you need to go into something that's more fast evolving, like cytochrome oxidase 1 or cytochrome B. So how does this whole process work from beginning to end? It starts out with a species list, and this is where you need to throw the kitchen sink at it. Every species that you think you could find in that environment that you are interested in should be in that list to include potential invasive species. The reason for this is once you get down to aligning and checking the primers, it can be difficult to go back. If the sequence isn't there originally, you can check and everything could work great. And then you throw in your invasive species that you weren't thinking about when you started the project, and your primers don't work. So you have to go all the way back to the beginning. So when you first start out, you need to be as inclusive as possible and include as many species as possible that you might be interested in. From there, it's going to checking the current databases that are out there, and CBI is the big one. You're going to want to look for your species of interest and see what loci are out there that you might be able to use. For fish, you're going to be looking for 12S, Site B, COX-1. For plants, you might be looking at MATK or TRNL or something like that. And it's basically a general search just to see what's available and what you could possibly design something off of. From there, you download the data, you align it, and then you go off of that alignment and look for places that you could potentially develop primers from. You eventually make your primers, and then you check it against everything on NCBI. So you initially develop your primers just off of your target species list, but then you have to go to NCBI and check it there to make sure that it doesn't hit some random other thing in your environment and give you a co-amplification and potential positive identification for a species of interest. So some of my markers work for, my 12S markers work for moose. So I could look for moose as well as all of my fish, but I had to make sure that if there was a moose in the water, I wasn't identifying it as some salmon species. And once you do that check, you can download the rest of your data from NCBI and create your own custom reference database. At any point in this process, if something goes wrong, you'll go all the way back to the beginning and start over and keep going. So this can go quickly, or it can take a lot of time depending on if the sequences are available or not. What you end up with is something like this. This is a Salvelinus primer set that I developed a couple years ago. It's in review right now. And you can tell there's a bunch of different Oncorhynchus species that it finds and a bunch of different Salvelinus species that it finds. And when you develop your primers, you're essentially at the same time developing your own custom reference database. And so once you make sure that all of your species are there, that none of them are identical, that none of them are matching, and they're different enough to be able to tell them apart, then you essentially have your primers and your custom reference database all wrapped into one, and you're good to go. Now, sequences are being uploaded every day, and you can be chasing goalposts through your entire project if you do this process on a continual basis. So what I typically do is start the project, check my primers if I've already developed them. And if I'm making new ones, say, okay, I made these primers on this date for these species. This is my reference database, and that's what's going to go into my metadata. So if new species are sequenced and put into the database later, they could be checked if you want, but you can go back and say, this is what I did at this particular time. And that allows other researchers to go and look at your data and validate your methods and make sure that what you're saying is correct. So one of the nice things about picking a large going with a multi-species approach is that for 12S, set of primers that I developed back in 2018, you can look at a lot of different species. All of these on this list, one are fish, but two, they can all be told apart just with the 12S primers. This is specific to Alaska, so it might not be applicable to your region, but these are a good, this is a good list for this particular primer set. Another thing is the added value. So for the 12S, when I checked a year later after I made the markers, I found that you could get 1,600 different genera, 3,700 species, and 420 or 4,800 unique sequences. And what this allows you to do is, if you're partnering with somebody else that has a separate project, you can look for other species. So you might have somebody looking for fish, but then you might have somebody looking for amphibians. Or maybe somebody wants to do a population analysis for moose to see if they're in the area. Generally, primers can hit a lot of different things, and it's a matter of checking to see if any other species are out there that you might be interested in. And this can take some time. So in the previous couple slides where I showed going back to NCBI to check your primers, this is what you would find, is that my 12S primers also hit some bats and a moose and a Dall sheep and a newt. And so it allows the opportunity to collaborate with other people at the same time, so it reduces your sample collection, sample processing costs, the time involved for everybody. And then once you have your samples, you can mix and match. And a lot of the samples that I get from the Park Service, they want to know what fish species are in a particular environment, but they also might be interested in finding if Alodia is present as well. So Alodia is an invasive up here, and it gets transported around by the float planes. And once it takes over a lake, it pretty much kills everything, and ADF&G comes in and nukes it, which we want to try to avoid. So part of a potential monitoring program would be, as people are collecting data for fish, you're also looking for Alodia. A paper I wrote recently was looking at diet of loons, the Gavia, and we were able to develop markers that were able to differentiate the 6 loon species as well as all of the fish in their diet. I'll talk about that in a little bit. But then I also made one for seagrass pathogens out in Eisenbeck, and you could mix and match those with the fish species to see what salmon are in the area. So there's a lot of different ways you can go with these markers once they're developed. Each of these would require their own first step PCR reaction. And then after you do that, you combine them all by sample, all the different loci by sample into one. And your second step PCR adds the indices to every single one of these markers for that particular sample. And you can end up with a ton of data. And as I mentioned, one of the problems that we found with Alaska is that there aren't a lot of Alaska-specific species information, genetic sequences up here. And so after finishing the paper in 2008, we decided to work on another project that was funded through NRPP to look at the mitogenomes of Alaska native species. And we're focusing primarily on Coregonus, Oncorhynchus, Salvelinus, and Cottus, and their 12S COX-1 and Site B genes. But one of the really cool things about this project is when you're doing a mitogenome, you start out with long-range primers. So you're looking at 6,000 base pair fragments, 4,000 to 6,000, which for a PacBio or a Minion really isn't that long. And so future applications of this would be, instead of looking at a short fragment that you would use on a MiSeq that's maybe 200 to 300 base pairs long, you can now extend that to a 6,000 base pair fragment that includes all of COX-1 and Site B, and then sequence that on a Minion or a PacBio or something like that. So some of the uses that we've done with these primer sets over the years, a lot of it started with fish, and you can see there's a lot of different fish species that are separated. You can do relative abundance through different areas, and you can see that different regions have different community structure. And you can see you were to plot this on a map, you could see how far apart these different lakes were. Another thing you can do is look at different sample types. So this was looking for eelgrass pathogens out in Eisenbeck Lagoon. And we looked at plant samples, sediment samples, and water column samples for these pathogens over time. And we found that there was a change over time, especially with Phytophthora gemini, where it seems to be a late summer species. And then this one was looking at scat samples from hunter-killed birds. So we took their hunters out in Eisenbeck all the time. And what we did is had collected the scat samples from them and then sequenced the scat for these pathogens. And what we found is that the pathogens make it through the digestive tract intact of these birds. We haven't figured out yet whether they're still viable and that's a potential mode for transmission, but it seems like it is possible. And that may be a reason why a lot of these sequences, what we found, are identical to European species of pathogens. And so it's possible there's hopping between Europe and North America and passing it back and forth. Another project we worked on was diet of loons, and the columns are split up by lakes, where it says Lake 1 is data that was collected by Lasky and Haynes, and then the scat sample collected from loons from their breeding ground areas. And this was done after the birds had left the area. There was no site of the bird actually defecating, so researchers went out there and basically just collected a whole bunch of scat samples and then threw them in a freezer in a -80, and I think these were about 4 or 5 years old before I got to them. And what we found is that basically the data lines up. The loons are eating what's in the lake, which is exactly what you would expect, mostly blackfish and stickleback. One interesting thing, though, is that they hadn't seen any Pacific loons up in the area. So if you look at Lake 6, that was a positive identification with 2 different markers for a Pacific loon. They had been known to be in the area. They'd seen them previously, but they hadn't seen them that season. But we were able to identify that particular loon species from a scat sample that nobody saw the bird from. So some final thoughts. eDNA is shiny. A lot of people are getting into it now. Metabarcoding is shiny as well. There's a lot of reluctance to get into it because there is a lot of bioinformatics that goes into it. But once a project is established and you've got your markers and your custom reference database, it really isn't that bad. But as I mentioned, one of the big things is that there's a lack of sequence data out there for potentially your particular species of interest. So the idea of working with taxonomists and museum curators to sequence those species is a high priority. And that's, I think, should be the focus for the eDNA community in general because of that lack of sequence data. And once we get that, then it's just a matter of being really specific about what you're looking for and the questions that you're trying to ask. Hopefully that was informative. Hopefully that answered some of your questions, and feel free to ask more.
Thank you, Damian. Just wanted to open up the field or open it up to questions. So feel free to jump in with questions.
Please do. That was a lot of information really quickly. So
Here, I'll start with one, Damian. So when we, most of the conversations we've had with this group and with many other, I guess, more manager dominated groups is a huge emphasis on validating the technique. So for targeted eDNA, we spend a lot of time thinking about sensitivity, specificity, and then, multiple stages of validation, beginning with the computer validation, so the in silico, and then progressing through the in vitro, and then finally the in situ. And when you gave your talk, it seems like you spent a lot of time thinking about the in silico, but kind of skip over the other types of validation. So when you're, discussing this technique with managers, how would you put kind of the reliability, repeatability, reproducibility, kind of confidence level with this technique? How would you try to convince people that this is good to go, or alternatively, nope, we're still really in an R&D phase and need a heck of a lot more work before we can actually apply this for decision making? Does that really long, mumbly question make any sense?
It does.
Okay.
And I know exactly what you're getting at because I completely skipped over it. Part of the in silico validation step is the running the gels. And so you do a lot of bioinformatics work, and everything works on paper, but you need to check it. And so that's when collaboration with other people, museum curators, whatnot, comes in. And what I do is if I develop a marker for a set of species, I try to get as many of those as possible, like physical samples, muscle tissue, fin clips, whatever, and run them individually with that multi-species marker, and run them side by side and make sure that they all actually work and they identify that species and run them on a gel. And if you can do that and show that you're finding all of your species with possible variations in there, then you can say, hey, okay, we checked it. It works on paper. We've checked it against tissue samples. That's great. And if you have a pond or an aquarium, my positive controls when I first started my fish was the UAF aquarium. And we knew what species were in there. And so after testing in the lab against muscle tissue, we tested against the UAF aquarium, and everything checked all the boxes and worked all the way down. So if you really wanted to get specific with a manager that was questioning whether these markers worked for a particular species, it's easy enough to just say, hey, I ran it against a species. Here's the gel. It works. There's a band. And it should be fine. That should be enough. And then if you do that with multiple markers, like I said, 12S, Site B, COX1, and you can say, we have redundant markers. They're all positive all the way through. That should be enough to convince them, I would hope.
Great. That sounds great. Nate, I see your hand up, so that either means you have a question or that you're not convinced.
Thanks, Adam, and thanks, Damian, for the presentation. Lots of good information and certainly a lot to think about. I guess my question is going back to, the importance of identifying the suite of species that you're interested in before you start this whole process. We're interested in going out and collecting a lot of samples and testing them for invasive species that we see occurrences in Utah, and so we're concerned about now, but we're also interested in the ability to have those samples archived and later tested a few years down the road for some of the invasive species that are in the region right now but not necessarily knocking on our doorstep. Is that doable, or is there something we need to, I guess, build into our thinking in terms of being able to expand the scope of what we're looking for down the road with these archive samples?
That is absolutely doable, without a doubt. The Loon scat that I mentioned had been in a freezer at -80 for, I think, 5 years before I got to them, and they were perfectly usable. The fish samples from the North Slope, or the eDNA samples from the North Slope, where we were looking at fish communities, those had been on Longmire for 3 years before I got to them. And so if you preserve them well enough, archive samples, museum samples are perfectly usable, and a great way to go back in time, essentially, and look for species that you didn't know you needed to look for when you first developed your project or were collecting the samples. So yes, without a doubt. I full-heartedly embrace and use Longmire buffer. I think it's great. I've used it for years now, and I've used samples. It was being used in the lab that I'm in before I got here, and I've extracted eDNA sequences from samples that have been on Longmire for over 10 years. We'd never published it because we didn't want to, but it's doable without a doubt, and it's a great way to go back in time.
It's
Great, thank you.
Just for context, Damian keeps talking about Longmire, so that's kind of how they're preserving the sample. You know, how your group, when you send samples, it's often like in silica or ethanol. They're preserving it in this Longmire solution, so that's kind of what Damian's alluding to. Lee,
Yeah,
you have a question? Or,
Yeah,
go ahead.
Ask about the other side of the coin, like potential sources of contamination in the lab. Is there a high concern? Like I work in the genetics lab at the University of Montana, and I have Dr. Allendorf's spin clips from 1991 sitting here. Is that like a huge concern, like more so than I would with my regular QPCR work that I'm doing?
Yeah, it can be, yes. What we have done up here at the Alaska Science Center is that we have a specific room designated for low copy ancient DNA and environmental DNA, and that's where we do all of our extractions. What they had found before I got up here about 10 years ago is that they were trying to figure out these communities and ended up getting a whole bunch of lamprey in their samples. Well, they had done a population genetics project prior to that, and there was lamprey DNA just floating around the lab, and it contaminated all their eDNA samples. So they moved it to a separate lab. If your samples have been capped and they haven't been opened, and even though they're in that lab, you could wash them off with bleach and alcohol, and they should be fine. But you know, be aware of other projects that were done in the lab, because it is a source of contamination. So in a perfect world, you would be doing your extractions and your sample processing in a separate lab from where you're doing your PCR thermocycler reactions and your MySeq prep.
Mm-hmm. Yeah, and that's how we have it set up now to do our eDNA samples in separate spaces. So cool. Thank you very much, sir.
You're welcome. And real quick, back to the Longmire buffer. So it's a tissue preservation buffer, but it's also a lysis buffer for people that haven't used it or are thinking about using it. It's great at room temperature. You don't need to refrigerate it or freeze it. You just cap it. I put some parafilm over the top of the cap to keep it from evaporating. And it helps with processing your samples. I can't remember the paper, but they compared regular extractions to Longmire, and they did it at an interval where they were able to see a difference after 2 weeks. So after 2 weeks in Longmire, they actually got better eDNA data from that sample than a sample that was just filtered and extracted. So I wholeheartedly endorse Longmire buffer.
Any more questions from the group?
I suppose I have a sort of in the weeds procedural question. So if you've talked to Adam recently, you've probably heard that we're going to be starting into some metabarcoding stuff. And one of the things that we are trying to do is sort of a platform comparison between Minion and MySeq. And our plan right now is to index our samples and then split those for the different library preps. I guess the thing that I'm unfamiliar with is whether or not there is anything, if there is a problem with that sort of workflow that I'm not seeing yet. In other words, can you get all the way up to the indexing step and still have the option of going to whatever platform you want to go to for the sequencing?
So
And
I
Damian,
haven't done.
Damian, can I interrupt for
Yeah.
a second? Just
Sure.
one of the goals of this group is to the education, and Patrick just threw out a lot of jargon. So as you are providing an answer, could you try to clarify, for example, what in the heck he's talking about with indexing, just to make sure everyone's on the same page?
Right. So the major platform that everybody uses right now is the aluminum MySeq. It's been around for a while. It's the workhorse. It does really good at short-ish fragments, 150 up to 600 base pairs. Generally, when you're looking at these sequences and you want to go to a manager, you want to have a really good sequence. So you do paired end reads. And what that means is that you've sequenced the forward direction and then you sequence the reverse direction. And because of the capabilities of the MySeq, you basically overlap those and say, okay, this is my sequence and we trust this sequence. The Minion is a newer platform. It uses nanopore technology where it basically slides DNA through a hole and goes through in sequences as the DNA strand goes through this hole. That's why they call it nanopore. There was a workshop last week put on by the nanopore people that was talking about the Minion, and they're getting a lot better. The problem with Minion traditionally is that it was going through the DNA was going through the pore so fast that they ended up not having a really good, reliable sequence. And so what people had been using the Minion for is to create a backbone because they can get longer sequences, 10,000, 25,000 base pairs easily. You would use that to see what the entire length of a sequence was. And then you would take your MySeq data and put that on top of that sequence to get the actual sequence that you were trying to find. So a lot of people use them in conjunction. Now, the Minion people, the nanopore people came out last week and said that their bioinformatics and their software is getting a lot better. And so you may not have to do a MySeq in conjunction with a Minion. I know they have developed multiplexing indices, but I have not run them yet on a Minion. So I'm not sure how they differ from the MySeq. So I don't know if you would actually go through all the way through adding the indices to your sample and then splitting it at that point. The protocol for the MySeq is that generally that two-step reaction, and I'm not sure what it is for the Minion. So to answer your question, you might have to stop at the extraction phase and then go through two separate simultaneous protocols to do a comparison. Yeah, that would probably be the way to do it. Not having gone through the Minion protocol. So sorry.
Thank you, Damian. I wanted to get back to Nate's question and just get your thoughts on, I guess, sensitivity of the technique. You know, when Nate asked this question for invasive species, I think that kind of presumes that your target out there is rare, low density. You know, from your experience, you know, what has been the sensitivity of this technique at picking up, you know, rare or lower density into the DNA from rare or lower density individuals?
So this is kind of a conceptual question because when you collect an eDNA sample, especially from a flowing body of water, you're taking a snapshot of that environment at that particular time, hoping to catch whatever you're looking for. If you're in a lake, you have to go through the different, you know, water levels based off of water temperature. So if you've got everything dialed in and you've got a robust sampling procedure, if it's there, you're probably going to find it. The hard part with invasive species is testing them. So as I mentioned, you want to run them on a gel to make sure that your markers work. And a lot of places aren't going to want you to send over an invasive species from someplace else so that you can do that work. So collaboration with other labs, potentially sending just the DNA as opposed to a tissue sample or an organism would be a way to do it. But once you've gone through all of those steps and validated your markers in silico and on a gel and potentially with a positive eDNA sample, and you use multiple markers and multiple samples, if you really wanted to get validation, you would rerun your samples 3 times. So in a perfect world, you would collect 5 samples at a particular location. You'd run multiple markers for your species of interest, and then you would run it 3 separate times. And if you did all of that and it was positive every single time, you're fairly certain that it's there. Now, that's not really feasible. A lot of people don't have the money to spend on running a sample 3 times or potentially collecting 5 samples. So you can subsample and repeat it 3 times or 3 subsamples and get enough data to say, yeah, we think it's here. This is why it hits multiple markers and multiple samples. So it just depends on how much convincing you have to do in terms of convincing the manager.
Thank you. Any other questions from the group? Or yeah, Nate, your hand's back up. Or yeah.
Yep.
Go for it.
So yeah, I guess a follow-up to that would be, suppose we're interested in looking for, let's just say, 8 different aquatic invasive species, but at the same time, we're also interested in looking for a handful of native mollusks that are threatened in Utah. From that standpoint, would we pursue that through QPCR, or is this approach better, given that we are looking for more than just, you know, a few species? It's more of a suite of species, even though we have that kind of defined. We're not really interested in the broader community beyond this.
Right. Once you get past a handful of species, QPCR starts becoming more expensive because you essentially have to run each one of those individually and then get your data. With a multi-species and metabarcoding approach, if you've got markers that can positively identify all of your species, it's just a separate first step PCR reaction. And I find it easier. It's been years since I've done QPCR. I generally don't do it anymore unless I absolutely have to, because I'm looking at communities. The hard part for that type of project would be developing primers that can find all of the mollusks of interest. When you start getting into invertebrates, you start having a lot of problems in terms of finding sequence data. And so that's when you have to go do the metagenomes yourself or find somebody else to do them. But if the sequence data is available, I think it would be a lot cheaper and more effective to go with a multi-species metabarcoding approach.
Great, thank you.
Yeah, Joe, go ahead and chime in. Oop, do I need to? Do you need me to unmute you? Or are you good, Joe? Are you? Try. Joe Crane, your hand is up, so you're free to ask your question, or take it down if you don't want your hand to be up. All right, well, maybe Joe does not mean for his hand to be up. I guess I wanted to open up the floor for any other questions or comments, and I also would love to encourage anyone else that has experience with metabarcoding. I know we have Katie Klimas on the phone, Kathy Richter, Catherine Abbott, Joseph Crane, and many others. So I encourage you guys to pipe in and feel free to add any responses to some of the questions if it's appropriate. Oh, Katie says her audio is not working well, but there are several metabarcoding assays for mussels. I know Katie is a mussel expert with the USGS, kind of in the Missouri Midwest area, and she's a great resource for anyone that has questions about mussels and metabarcoding. Kathy, go ahead.
Yeah, so I have a question for Damian. In our experience, when we use multiple different primer sets, we often get different sets of species from each one. Usually there's some overlap, but not complete overlap. And we've tended to interpret that to mean that, you know, the entire set of species that we're detecting is present, even if we're only picking it up with one marker. You know, we make the analogy that we're like fishing with two different nets because each net is better at catching different fish. So I wonder what your take on that issue is.
Absolutely, yeah. When I first started, I was focused on 12S for some reason, and we're getting a lot of species with those markers, and it worked really well. But the problem was when you tried to differentiate the different salmon species or cod species, it didn't work out. Generally, the markers that I develop for metabarcoding get all of my species of interest. And if I can't get them with one marker, I'll develop another. So I've got Cox and Site B for Codus and Oncorhynchus and Salvelinus. And so if I can't get something with one marker, I'll make another one. And that's how I get around that problem.
Great. Thanks, Damian. Joe Crane, I noticed you had a hand up and then you left the meeting, then you're back in. Did you have a question?
Yeah, you also couldn't hear me cuss about Microsoft Teams not being
I could
very
feel it.
helpful. Yeah, I felt that one too. Sorry about that. I just wanted to weigh in on the kind of the economics and the trade-offs on the NGS versus QPCR for innate a little bit. That some of the questions that you need to ask are not just how many species you're looking at, but in what taxonomic groups they are. And for us, you know, essentially that breakpoint is 2 species within the same taxonomic group. It's more or less the same. It can cost to run QPCR as NGS. And then once we are at 3, then typically it's better to do the metabarcoding approach just from a financial standpoint. That being said, there's also questions about false positives and false negatives. So with QPCR, are you more likely to detect something if it's there? Typically, the way that we do it, we have pretty similar sensitivity for QPCR and NGS if we run enough replicates. It takes a couple of replicates, like we do QPCR is 3 replicates, for NGS often it's 6, just to get somewhere similar. And then, you know, empirically, some of the work that needs to be done is really starting to get at effort detection curves or effort diversity curves. So how many samples from a site do we need in order to detect 50%, 85%, 95% of the species that we know or think to be present? And that's kind of work that still needs to be done for lots of different locations so we can get a feel for it. We've done it at a couple of different sites, but not enough to have any generality. So economics is one thing, just as Damian had talked about, and he's right, once you get past a couple, then it's definitely better, all other things equal, to go to NGS. That being said, all other things are not always equal. So there's a couple extra calculations to do in there.
Thanks a lot, Joe. That was a great explanation. Any other questions or comments? Great. Well, Damian, thanks so much for your time. I hope to God that this recording works, and if it does, I will do what I can to get it posted and shared. So until that time, I guess we will talk next month. And Catherine Abbott, even though you were late on, I'm going to ask you to be on the hook maybe for next month to report back about the DFO workshop for next week, if that's okay. I'll put that in your queue.
Okay.
Awesome. Great. Well, I hope everyone has a great holidays, and we'll be back together in a month. Have a great rest of your day. Thank you.
Thanks for coming, everybody.
Thank you.
Thanks.
Thank you.